Well, welcome to part 2 of DNA profiling. We may have a little bit of corridor noise here. It's break time, and of course the younger kids are running around making a lot of noise, but we'll forgive them that. And hopefully the whine that I noticed on the first video isn't too pronounced on this one. Who knows? We will see. So we left our last video thinking about the differences in our DNA, differences between different people according to how many STRs they have, short tandem repeats. We're now going to think about how we can use that to give someone a DNA profile. We're going to talk now about something we're going to come back to later on in this video, and that is something called gel electrophoresis. Gel electrophoresis is this process you can see in front of you on this screen here. And in gel electrophoresis, we can separate fragments of DNA according to their size. Now, DNA has this structure here with its bases paired up in the middle, and then with a sugar, deoxyribose, of course, in DNA, just ribose in RNA. And then it has these phosphate groups. Well, phosphate groups are able to donate H+ ions and therefore leave a negative charge behind them. The fact that they donate H+ ions means they are acidic, hence these are called nucleic acids. And the fact that they can donate those H+ ions and leave negative charges behind means that DNA is negatively charged. It further means that if you put a voltage across a gel and you load up DNA samples at one end of the gel, those DNA samples will migrate towards the positive electrode. And they'll migrate towards it according to their size, with the smallest ones moving faster. So fragments of DNA can be separated according to their size, something we will revisit later on. Well, what do we do next? We cut up our chromosomes. Here we have a pair of scissors next to a strand of DNA, and yes, you will remember these from genetic engineering. They are restriction enzymes which do this. So here we have chromosome 9, and we cut it up. Now, these restriction enzymes, you can see this structure here is the restriction enzyme, and you can see it wrapped around a strand of DNA. They cut DNA at specific sites. So, for example, here might be one cut site. Whenever this particular restriction enzyme finds this sequence, it will make a cut. Note, it leaves unpaired bases after making this cut. Those are called sticky ends. That will be very important when we come to genetic engineering later on in the A2 course. Different restriction enzymes cut at different recognition sites. That's not a particular surprise. They have different shaped active sites and therefore recognize different base sequences, because different base sequences will be complementary to their active site shape. So we end up with lots of different cut sites. Take this example of a chromosome. Echo R1, that refers to a particular restriction enzyme. The haploid genome of the human is 3 billion base pairs. Therefore, these cut site sequences will occur at random throughout our genome. Therefore, if you apply a particular restriction enzyme, you will cut your genome into fragments of different lengths. So, for example, if you are using the restriction enzyme Echo R1, you'll have a cut site here and a cut site here. The next cut site is here and there. And you see you get fragments of different lengths depending on where those cut sites have been. This would be a very short fragment between these two, this a longer fragment between these two here, and a much longer fragment, we assume, going before this first cut here. So we have means of chopping the human genome up into different fragment lengths. Why might fragment lengths be different between different people? Well, go back to thinking about those STRs, short tandem repeats. Different people have different numbers of STRs. Therefore, this will change the length of DNA between cut sites. Let's say between this cut site here and this cut site here, just next to it, one person has 4 STRs, another person has 6 STRs. One person's DNA fragment here in this section will be longer than the others. Therefore, their fragment lengths will vary from person to person according to the number of those STRs. Another thing that may occur is that one person may be carrying a mutation from the normal human genome. Take this Echo R1 site here. This is our cut site. This is the sequence Echo R1 is going to recognize. These triplets code for these 3 amino acids, ACG for this, AAT for this, TCC for this. Now, AAT codes for ASN, AAC also codes for this amino acid ASN here. So actually, this mutation does not affect the protein produced. However, what it does do is it removes the Echo R1 cut site. It removes it because Echo R1 will not recognize ACG, AAC, TCC as a cut site. Therefore, your mutation has gotten rid of an Echo R1 cut site. Therefore, if we consider our chromosome again, let's say, for example, this mutation occurred here. This would mean our cut site at this point would be removed, and our fragments would now run from this cut site all the way up to this cut site, changing the fragment length between one person without the mutation and one person with the mutation. So there are different ways we might get different fragment lengths. These different fragment lengths are called RFLPs, restriction fragment length polymorphisms. Polymorphism is just a word really meaning different alleles which are present in a population. So we end up with our cut, after our cut from restriction enzymes, a large number of different lengths of DNA which will vary from person to person. Now we get on to our gel electrophoresis. We chop someone's DNA up using restriction enzymes, and then we run a gel. We can then visualize the DNA bands that are produced by this. For example, we might use fluorescent nucleotides when we do it. And so we just shine a light on it and up come our DNA bands. Or maybe we stain it with an ink which specifically binds onto DNA. Or maybe we use radioactive nucleotides. That would be a more old-fashioned way, and obviously one more fraught with difficulties for health and safety. Finally, we can look at our results. We will get a unique pattern of RFLPs. You will have your unique pattern of RFLPs. And these patterns differ between individuals and are going to be unique to you. Unique to you. You will have elements of it which are identical to either one of your parents, but you will have inherited different chromosomes to your siblings unless you and your sibling are monozygotic, twins, triplets, etc. Therefore, you can use this in crime forensics and paternity cases. And in particular, you may have heard of this particular case. This is Mr. O. Simpson. You may know him as O.J. Simpson, and N. Brown, R. Goldman, people he was cleared of murdering, but then was prosecuted for murdering on a civil case. And I'm sure this played a very important part in that case. Well, thank you for listening. Make sure you go back and re-listen to material so that it's absolutely clear in your mind, and do take notes down the side of those pages. Thank you.